Welcome back to the Vision Symposium. So now we're going to let Leo Fleishman tell us all about how visual system light environment interactions shape the evolution of diversity in anolis lizard signal colors.
Thanks all for coming. So the first thing I wanted to say is just quickly why evolutionary biologists should care about signal diversity. And the main reason is if populations evolve different signals, that can often lead to breaking of gene flow and lead to speciation. So it's a really fundamental question what the causes are of signal diversity. So that's what I've been looking at. The sort of conceptual framework I'm going to talk about is I'm sure you've heard of it, it's called sensory drive hypothesis. And the idea of sensory drive hypothesis is you have some critter living in some set of conditions and it evolves a display that's optimally visible. And then some piece of the population moves to a new habitat, and then there is evolution of the visual system in response to that habitat, and that favors a new signal for optimal visibility. And then at some point, if they get back together, they've got different signals, they may not be able to interbreed. Just one thing to point out is you don't have to suppose a change in the visual system. It could be simply the animal moves to a new habitat, and the new habitat favors a different signal. And that will be important for this study. The majority of really cool examples or clean examples of sensory drive seem to come from aquatic systems. And I think that's because water has a real strong filtering effect on light, and it creates these backgrounds, and air just doesn't do that. And so the examples from terrestrial systems are rarer. So those of us who work on terrestrial systems, we have fish envy, so we keep trying to find a good example. And so I've always thought, I've been working on this for a long time, that this was sort of the perfect example, the perfect place to look for sensory drive in a terrestrial system, because you've got these anolis lizards. They signal with these colorful throat fans, colorful dewlaps. They only use it for signaling, so you don't have to worry about crypsis or possible confounds. They live in different light environments, mostly because they thermoregulate, and also they occupy different sort of vegetation structures. And there are lots of species, and in many places there are lots of species sympatrically. So they're all sort of, it would seem to be a great setup for a sensory drive example. So the first step in this kind of study is we've got to understand the visual system, in particular the color vision system. So for many years, I would make my way over to Cornell University and sit in Ellis Lowe's lab, and we looked at a lot of lizard retinas. And this is an anolis retina, kind of squashed down. And the only point is it's got lots of cone photoreceptors, and each of the photoreceptors has an oil droplet filter. And in the outer segments are different pigments, and this is the underlying basis for color vision. If you look at these things in more detail, typically this is what we found: 4 kinds of cone photoreceptors, 4 classes: a long wave sensitive one, middle wave sensitive, short wave, and then a UV sensitive cone. And the graph on the right just shows the typical the absorption curves or the spectral sensitivities of the 4 typical cone photoreceptors. So Ellis and I tackled a whole bunch of these using microspectrophotometry principally. And the only reason I'm showing you this list is, so this is the 17 species that we looked at, plus 17 anolis plus 1 outgroup. And if you can see it, which I don't know if you can, they come from a lot of different clades. There's a lot of different island sources and a lot of different clades. And the habitat types, which are listed in the far right, are quite variable. They vary from full shade, partial shade canopy, full sun, et cetera, and they're mixed up nicely in the clades. They're not like one clade is all full sun. It's all mixed through. So we have lots of species, different evolutionary backgrounds, and lots of different habitat types. So it seemed like a good place to look for differences. So this is sort of the outcome of those MSP studies. On the y-axis, we have what are called the lambda max values, the peak absorption of the different photoreceptor types. On the x-axis are the different species. And I hope it comes across as they're all pretty similar. There is one weird outlier at the end, number 17, is Anolis carolinensis, which for some reason is very redshifted. Happy to talk to people about that if they're interested, but I'm not going to talk about that right now. The other 16 species, pretty well behaved. They all have pretty close to the same sets of photoreceptor pigment values. The UV is missing in a few species, but the UV is the rarest of the cones, and we didn't have a lot of individuals of some species. I'm pretty sure that's just a sampling problem. So basically, for functional application of color vision, they're all pretty much the same. In fact, we've modeled whether the small differences we see matter, and they really don't. So basically, they're all sharing pretty much the same visual system. So we know that anoles from widely different habitats and different clades share the same basic pattern of photoreceptor spectral sensitivities. So now we go on to the second part of sensory drive. Do differences in habitat light alone favor different dewlap colors? And one thing I need to point out is we've done a lot of physiology and behavior experiments on signal visibility. And signal visibility is related directly, correlates well with chromatic contrast. And you can measure chromatic contrast in different ways, but it's basically a distance in perceptual space between a stimulus and the background. Gives us a nice prediction of the visibility of the signals. So we know how color, signal versus background colors affect the visibility of these signals. And one thing we have discovered is when you're in a green world, like this guy is, red is the most conspicuous color. It stands out the best. We can model that. We can measure it behaviorally. It's true. So we then, next step is to look at the different habitat types. We've looked at quite a lot of them. I'll show that later. But for now, I'm just going to talk about a study we did of 4 species from Puerto Rico. These are sister species, but we have one who's out in the full sun, who likes it really hot and no shade. We have 2 partial shade species, one that's kind of likes forest edges and little clearings, and then we have one who likes dry forests, Christotellus, and then we have a species from heavily shaded forest understory. So we have a nice, across these 4 species, a nice range of different light habitats. And the way we measure light habitats is pretty simple. You get a willing undergraduate slave, and you walk along habitats. There's a lot of lizards in these habitats in Puerto Rico. You just walk along till you see one, and step back a few meters, wait till it displays. Then when it displays, we run up and measure the light at the display location. And what do we measure? We measure spectral irradiance, spectral and total irradiance, sidewelling irradiance, because we want to know the light lighting up the dewlap in the eyes. And then we put on a little filter and measure the background radiance, a smaller angle measurement of what the background looks like. And that allows us to characterize the light and the habitats. We also bring the lizards into the lab and measure spectral reflectance of the dewlap and also spectral transmittance. It's really important. These things transmit a lot of light. They're thin. And to characterize the color, we need to know the transmitted light as well. So we measure both of those. And one useful thing about that is once we have these measurements, we can plug that dewlap into any set of field conditions with those irradiances and radiances and calculate the relative visibility of different dewlaps. So what do the habitats look like? Here are, these are normalized spectra from the different habitats. On the left, we have irradiance spectra. You look at the irradiance spectra, there's a little bit of difference in one of them, maybe. The green one is the forest, and it's a little greener than the others. The others are a little broader. But in truth, color vision systems go through chromatic adaptation. So if that's the light illuminating their eyes, they're going to adjust to that, and it's going to have really no impact on the color appearance of objects in there. When we look at the radiance, the spectral radiance, it's even more consistent. They're almost identical. And again, this is relative. These are normalized spectra. They live in a green world, and most of when you average out, what it looks like is you have a green background. So we find little difference in the color properties of the different habitats. We find little difference in the color vision of the different species. So we're forced, that I was some years ago, to say, is there really any relationship between dewlap color and habitat light? And well, there's one other aspect of the habitat that's kind of important, which is the total intensity. So here we have the irradiance values from the different habitats of the different species. And you can see it's on a log scale that there's about an order of magnitude increase when you go from the dark forest on the far side to the partial shade to the full shade. So there are big differences in total intensity. Why does that matter? Turns out when you get, especially to low light, it starts interacting with chromatic perception. So here's an example. Hopefully the square on the right, the red looks very vivid, the yellow looks vivid too, but when we darken the whole thing, the red doesn't look so good anymore. It doesn't look very red. It doesn't stand out very well. In fact, you can model it, its visibility goes down, but the yellow holds up pretty well. So we thought that's interesting. So we did some experiments like this. In this experiment, the lizard sits on top of a little perch in a little cage. The front of the cage is glass. He's looking out at a camera with one eye. In his visual periphery, he sees this checkerboard of green squares. And then we introduce a color, in this case, in the top one, we introduce a yellow of same brightness to the square it's replacing. And we just watch to see whether he shifts his gaze and looks at the newly introduced yellow square. And we do lots and lots of these with lots of lizards, and we generate probabilities of detection. So we did this with yellow, and then we did it with red. And we also did it under low and high light conditions, representing sort of roughly the different habitat conditions. And sure enough, under the light conditions, red is the strongest stimulus. It gets the most responses. But when we use a darker condition, more equivalent to sort of forest shade, red drops, and it drops below the yellow. And the yellow has enough light coming off it that it's not affected by these changes in light intensity, but the red is. So red is best under bright light, but it drops behind yellow under darker conditions. So this is sort of shown here photographically against in a bright light condition against a green background. A red dewlap really shows up, but when you start getting into darker conditions, the red doesn't show up so well. And in darker conditions, actually yellow shows up better against these green backgrounds. And again, we can look at it and model it and measure it behaviorally. So one way to measure redness or yellowness of these dewlaps is to look at what we call cut-on wavelength. It's just the point where the reflectance goes from a low to a high value, and the further that is to longer wavelengths, the redder the dewlap looks. And a nice way to measure red and yellowness in dewlaps. So we looked at 17 species again, and in this case, we plot dewlap cut-on wavelength versus habitat light intensity. It's on a log scale, and you can see that in the bright conditions over to the right of the graph, we have lots of red and orange dewlaps. We go down to the dark light, we have lots of yellow dewlaps, and these correlations are significant, either corrected for phylogeny or not. We have highly significant correlations. So, and there's one other small trick, which is that red dewlaps have denser pigment than yellow or white dewlaps. So if you don't have red, if you have yellow or white, you get a really nice, a lot of light coming through the dewlap. It really lights up. So that's another thing that adds to its visibility. And this is just a cute picture from Manuel Leal in Cuba. There's the transmission with the light happening to catch it. So that's another advantage of being white or yellow. So I concluded the visual system has influenced the evolution of different dewlap colors in different habitats because differences in habitat light intensity alter the perception of chromatic contrast. So we actually do have a sensory drive effect favoring the evolution of different dewlap colors. But what happens, it's not simply based on the chromatic properties of the environment. It's a combination of the chromatic properties and the total light intensity. Thank you.
So, next talk right now is Parker Hughes telling us about overturning a century of thought, evolutionary patterns of pupillary light responses in animals with quote-unquote immobile pupils.
Yeah, thank you, Dr. Longhorns, for the awesome introduction. So we can get right into it then. So as we've seen from many of today's talks so far, the diversity of visual systems is just as diverse as life itself. And while these eyes vary remarkably in their morphology and complexity, the fundamental purpose of an eye is to gather and filter light and channel it back to the retina to create an image that maximizes sensitivity and acuity in order to allow the organism to conduct visually guided behaviors such as foraging, predator detection, navigating social situations. And briefly, I just want to establish a simple definition of a few terms for sensitivity, resolution, and acuity. So for the purposes of this talk, sensitivity is just the ability of an eye to gather and focus enough photons to the retina to create an image. Resolution is just the ability of an eye to then distinguish fine details within that image, and then these two marry together to become acuity. And so you can see this image on the screen. It's the same scene 5 times over, but as more light is gathered and focused onto the retina, the image becomes easier to create and resolve. And so organisms have many different visual needs based on their environment and ecology, and selection can act upon these needs to drive the adaptation of the visual system. So take these two eyes up here, the human eye and then the fish eye. You can see that the human eye shares the same overall structure of the ancestral fish eye, but there's some key adaptations such as a thicker cornea and then a smaller and altered lens shape That way it can successfully and adequately gather light through the air, while that fisheye still has a really thin cornea and a large spherical lens to gather light through the water. And so while these different structures can adapt to these different environments, there's very few environments on Earth where the ambient light level stays the same over large periods of time. And so for any given eye to actually be able to adjust to varying ambient light level would suggest that it would have a much higher visual performance. And so take these cat eyes here, for example. I'll see if this will work. Here on the left, you can see in bright light situations that cat eye pupil width will shrink down to just a sliver. That way it can highly restrict the number of photons getting back to the retina in order to create an image that isn't too bright to resolve. And then that same animal can also have a quite significantly larger pupil for dark light conditions in order to gather enough light to create an image. And so this kind of pupillary light response is seen in a variety of taxa, from our own eyes to eyes of raptors and nocturnal species, and even in the lensless pinhole eyes of the nautilus, which is pretty interesting. And while we're really familiar with a lot of simple circular pupils that may expand or constrict, many taxa, such as like this skate on the screen and other elasmobranchs, show some serious variation and creativity in their responses. But what I found is even stranger than this odd response right here is that a large group of species, teleos fishes, are thought to lack not only a pupillary light response, but pupil mobility in general. And this is extra weird just because this group of ray fin fishes makes up about 50% of all extant vertebrates on Earth. And given that they have similar taxa to skates and things, where they have that spherical lens, as well as the obvious advantages that PLR offers in visual performance, it just doesn't really make a whole lot of sense for them to lack this ability. And so while you should not and do not need to read this table up here, I just wanted to show that this is not just me saying that this is a thing seen widely throughout science, but it's largely found in the literature. So this is just 18 different sources that we've found over the past 80 years or so, stating that teleos do not have mobile pupils for one reason or another. And so based off of all of that, this study aims to address two major questions. So the first being, just do teleos pupils respond to light, or is it true they have fixed pupils? And then the second being that if these fish do have pupillary light responses, what are the ecological mechanisms driving its evolution? And so here, for that first question, we did two major tests to find out what's really going on with these guys. The first was just an exhaustive literature review, really just to understand the current and historic study of these abilities and systems. And then the second was we just tested these fish directly in a standardized manner and recorded these responses with a camera and an IR light while exposing them to different ecologically relevant light levels. And so for that literature review, it exposed actually a very interesting history in the science behind pupillary light responses. So in total, PLR was described in at least 16 orders and 51 species, with 27 of these species being described over a century ago. And these span from incredibly basal clades to highly derived clades, as well as the PLR of many non-teleos that I have here up on the screen, such as lamprey and sturgeon and elasmobranchs, had their PLRs described over almost a century and a half ago. And we're not really sure what happened to where these outgroups didn't ever have a loss of understanding and teleos were like left out of the club for some reason. But this makes it kind of like extra strange. And so then we took that evidence of PLR within this group and directly tested a wide variety of model species that we selected not only to give us a pretty representative sample of all of teleos for the most part, but as well as really important ones such as Medaka zebrafish, stickleback, and like cichlids and such that are not only used a lot in ecoevolutionary studies, but across like a variety of different disciplines. And we predicted that not only will they all have it, but it's likely widespread across the group and ancestral. And so this prediction was supported. And so far, I actually haven't had a single species without some sort of PLR. I don't know if I have time for this, but I've even tested cave mollies that are like 13 pools deep within the caves down in Tabasco, Mexico, and they have it, and there's like papers about them actively devolving eyes. So you'd think if anyone was not going to have it, they'd not have it. Anyways, I probably don't have time for this. So onto the next part of the study was trying to now figure out what's going on with these guys. So they had a pretty wide diversity and variation in their responses. They were not all the same. So like cave mollies didn't have a super strong response, but they still had one. And we found that in many studies of visual ecology, variability of light environment drives the evolution of the visual system. And we think that this is probably going to extend into this study as well. And so like I said, there's all that variation across these species. But if we look at one specific species, actually our model species, Gambusia hubzai, we can really start to nitpick about these ecological drivers. So this system is just a part of a post-Pleistocene radiation of fish. They live in blue holes down in the Bahamas, which are just quickly, they're vertical caves that filled with water about 10 to 15,000 years ago and were colonized by these guys. And it just has created this amazing natural experiment, highly replicated, allows us to tease apart these mechanisms behind complex trait adaptation. And so this is just a representative trial of a Gambusia hubzai, illustrating a pretty strong and distinct response. Let's see if this will work here. So you can see in our initial control light here, they had a pretty like normal pupil, I guess. But then as they got exposed to more light, it quickly constricts and then dilates back down with reduced light levels and then dilates further when they're in the dark. And it's pretty cool. So you can actually see these visually, like just looking at the fish as well. And so this was a snapshot taken of one of our hubzai trials while the fish was exposed to about 500 lux, or our control light level. And so try and pay particular attention to the size of the pupil relative to the rest of the eye here, as I'm about to show you this exact same fish, exact same trial, but at 0 lux. And so I wrote down actually what I said when I first saw this. Whoa. And then, so you can see that the pupil here has clearly increased and dilated, and conceptually this should increase this fish eye's ability to gather light in dim and dark conditions. And so what exactly can influence the light environment of these fish? So the first factor that we hypothesize to be of particular importance is turbidity. So you can see here this first blue hole, this is Ken's blue hole. It's not very turbid, has pretty clear blue water, really nice, limited amount of material suspended within it. And then this blue hole here is actually named murky brown blue hole, which I'm sure is fairly evident as to why. And so it has a really high level of dinoflagellates within it. And due to this, the water is much more turbid and discolored, and it's not very fun to swim in. And this causes much more rapid light attenuation with depth and distance. And so we predict that fish should evolve stronger PLRs, particularly a greater ability to dilate their pupils in order to gather more light. And so another amazing system about these blue hole systems is they have rather simple fish assemblages, with some of these blue holes being just solely inhabited by Gambusia hubzai, and then others being inhabited by Gambusia hubzai, and then a predator specialist of theirs, the big-mouth sleeper. And so you may be kind of wondering, like, what does predation have to do with anything about light environment? And so what essentially happens is in these low predation blue holes that we have kind of pictured here, that's just a bunch of hubzai around some stalactites, is these fish have free reign to go wherever they want within the blue hole. So they'll go in open water and shallow nearshore environments that are really bright. They're active at night. They'll go pretty dark, down really deep. We've observed them deeper than 30 meters. They'll go in and around complex habitats like these stalactites on the screen. And they'll go even inside and outside of caves, kind of at a moment's notice, creating a super dynamic and variable light environment that they're experiencing, and just kind of an overall just really random light environment depending on where they're at in the blue hole. And so this is quite the opposite from a blue hole that does have big mouth sleepers within it. So this is a very similar situation, but from a high predation blue hole, and the only fish you can see here are actually just the predators. And so because of this threat of predation, the fish are really restricted to shallow near-shore environments and haven't really gone deeper or been observed deeper than the top 2.5 meters of water. And they just live in a much more homogeneous light environment. They don't have any activity at night. They don't really use complex habitat. They're not going in caves. They're not going deep. And it just creates a much less variable light environment, many fewer light environments they're experiencing at any one given time. And so in our lab, we maintain 8 different populations, 4 from high predation blue holes and 4 from low predation blue holes that span a gradient of turbidity. And I tested F2 males and females from all of these. And so not only did we find results that supported our hypotheses, but we found that these responses were not simply better or worse pupillary light response, but more directional in aspects such as dilation and constriction. We analyzed our findings to compare multiple potential ecological factors via model selection and model averaging. And we just found that turbidity and light environment via predation regime were the most significant things. That's kind of why we focused on this for the talk. But we did test other factors such as resource availability to see if competition had any influence on the evolution of pupillary light response. And just didn't find any significant findings there. Let's see. And so we kind of found like low predation blue holes, likely due to that very variable light environment, evolved greater constriction abilities. And then fish inhabiting blue holes with higher turbidity levels evolved greater dilation abilities. And so we analyzed these results also with both multivariate PCAs and univariate analyses, and the results really didn't differ at all. And so in conclusion, we found that teleost fishes can and do utilize pupillary light responses. It's likely ancestral and just as likely that virtually all fishes will show some sort of PLR. This actually goes against the current consensus in the field of visual ecology. And moreover, we've uncovered some of the potential key ecological drivers driving the diversity of these abilities. And we did have some interesting aspects that I just kind of wanted to touch on here in sexually dimorphic abilities. So in low predation blue holes, females specifically evolved greater early bright constriction, so first 30 seconds after exposure to bright light, while males have greater average and total constriction abilities. And while like that's a little strange, the overall theme here is low predation equals greater constriction. We're still trying to figure out what's going on with these things. And then a similar thing was seen in turbid environments. So higher turbidity females evolved a greater early dilation response, while similarly males evolved a greater total dilation response. And just as like that's also kind of a work in progress now, the theme overall is that turbidity evolves better dilation abilities. And so really quickly, I want to acknowledge all of our collaborators who have either helped out with this data collection or sent me fish to test, as well as SICB for funding part of this study. And I think I have some time for questions.